Hi A&P2 students. This is a video over the urinary system. We're continuing on. The last couple of videos we kind of focused on the process of glomerular filtration that was happening at the renal corpuscle of our nephron. So the next few objectives, starting with 20, we're going to focus on the tubules and what processes are going on there, which you should know are tubular reabsorption and tubular secretion. So the first two objectives here, we're going to talk about the reabsorption of substances in the blood that get completely reabsorbed, like we want to reabsorb all of them and not let any of them enter into the urine. So we're going to focus on those first, and then we'll go on to some substances that their reabsorption is regulated, so not all of it's going to be reabsorbed, but varying amounts will, depending on what the body needs. And then we'll end with, this kind of goes on the next page, then we'll end with the waste products and how they are processed within the, how they get into the urine, and we'll talk about that process too. Okay, so first we're going to talk about substances that are completely reabsorbed. So remember our nephron here. Remember, it's not anatomically correct, but it's easier to look at it this way and talk about these processes. So we have a renal corpuscle here, our afferent and efferent arteriole coming in there to the glomerulus, coming in and leaving. Proximal convoluted tubule, loop of Henle, distal convoluted tubule, collecting duct, and then the collecting tubule, and then collecting duct. So when we looked at the picture before, kind of just went over the basic definitions of tubular reabsorption and tubular secretion. We just kind of saw the little peritubular capillaries running right next to the tubules. So we need to understand reabsorption and secretion, understand a little bit better, we need to kind of zoom in and see what the structure is of these two parts. And this is, it's kind of hard for me to get it all in there. I guess we'll do it that way. So this is, you can just see like a little bit here of our renal corpuscle or little glomerular capillaries. So we're looking at, and this is our beginning of our proximal convoluted tubule. So you'll notice that we have this little lumen where the tubular fluid is flowing, and then the wall of the tubules is lined with these little epithelial cells, and they're simple cuboidal ones. So that's what's lining them. Now, if a substance wants to move or needs to move from the tubular fluid and get back into, this is our peritubular capillary here, then what it has to do is it can either go between the cells, so that's a possibility. A lot of stuff though will pass through the cells, and so this is our little protein carrier. There's one there, there's also one over here. So they'll have to pass, if they have to pass through that cell that's lining the tubular cell that's lining the tubule, it'll have to go through this little protein channel, get into the cell, and then it'll probably have to pass through another protein channel to get out. And then what it does is it gets into the interstitial fluid. So these two basement membranes, two structures, are not fused together. There's actually the interstitial fluid between them. So it's a little bit different than the respiratory membrane where the basement membranes were fused together. They were much closer to each other. So in this case, we don't have a real close touching going on between these two structures. So there's a little bit of interstitial fluid here, so the substance will have to get into the interstitial fluid and then pass through the wall of the peritubular capillary to get into the blood, back into the blood. So that's what substances are doing. If they're moving from the tubular fluid back into the blood, we call that tubular reabsorption. And then we'll talk about some substances that move from the blood back into the tubular fluid, and so that's tubular secretion. We're going to focus on tubular reabsorption for now. All right, so that's just a little overview picture. We want to talk about the substances that are completely reabsorbed, and so the two main ones that we're going to talk about are glucose, and this could be other nutrients too, but we kind of focus on glucose. And then proteins. And I know that we talked earlier about how the proteins don't get through that filtration membrane because they're too big or they have a negative charge, and that's true. But sometimes little proteins, intermediate sized ones, can get through. And so we will have a process where they get reabsorbed and back into the blood. So glucose and the proteins, those are two things that would be abnormal in the urine. If our kidneys are working properly, then we should be able to reabsorb all the sugar, all the glucose, and all of the proteins. So they should not be in the urine. And those are things we need. Our body cells need those. So we don't want them to they're not waste products. We don't want them to go into the urine. So, all right. So for the glucose, we're going to go down here. There we go. All right. So how glucose works, and they've just kind of I'm going to make this a little smaller maybe so you can see it all. So up here is our peritubular capillary, and then we have our interstitial fluid, and then we have one of these tubular cells. And again, we're still in the proximal convoluted tubule. So remember, most of the tubular reabsorption that's going on is going to be going on in the proximal convoluted tubule. So that's where most reabsorption is going to happen. So be aware of that. The glucose, once it's in the tubular fluid, it has to move into a cell, and it's going to be going from an area where it's in low concentration to high concentration. So they'll use this little secondary active transport protein. So sodium's going down its concentration gradient from high to low, so the glucose will go along with it. And then once the glucose uses that little protein pump to get in, then it's going to need to move through the, they call it basolateral membrane, so just kind of the membrane at the base of the cell. And it's going to have to use another membrane protein. Now, in this case, it's going from an area where it's in high concentration to low, so it just diffuses down its concentration and gets into the interstitial fluid, and then it'll move directly into the blood after that. So that is going to happen in the proximal convoluted tubules, and if all is working properly, body's healthy, kidneys are healthy, 100% of the glucose gets reabsorbed there, because we don't want to lose any glucose because our cells need it for energy. So even if they don't need it right now, we're going to store it somewhere else. Okay, so that is the overview of glucose reabsorption. Now proteins will go through this process too, where they'll be in the tubular fluid. They're going to pass through the tubular cell, through the interstitium, and get back into the blood as well. I didn't have the details of how that works, so just be aware that small proteins, intermediate proteins will get it and get back into the blood at the proximal convoluted tubule, and they should, if everything's working properly, all the proteins should be reabsorbed 100% there. All right. So that was just a discussion on explanation of the reabsorption of nutrients such as glucose that are completely reabsorbed. All right, the next objective is over. This is an area where we talk about the concept of transport maximum. So because our glucose, in this case, because it has to use a protein to leave the tubular fluid and get into the tubular cell so it can make its way back to the blood, there's only so many of these transport proteins associated with these tubular cells. So we have a maximum amount of the glucose that could be reabsorbed, because if we have the maximum amount of glucose in our tubular fluid, then we would saturate, we say, like all these little transport proteins would have a glucose that they're moving across the membrane, and they couldn't move anymore. And so that means if we have more glucose than that maximum number, that means all the glucose that can be reabsorbed will be reabsorbed, and all those little transport proteins would be saturated, meaning that they're completely bound by the glucose. But that means that there could be the extra glucose that could be in the tubular fluid will not have a way to get reabsorbed, and it'll just keep flowing down the proximal convoluted tubule and make its way into the loop of Henle and onto the distal convoluted tubule. And here's the thing, there's no other, only in the proximal convoluted tubule do you have these little transport proteins that can reabsorb glucose. So once it gets past this point, you can't reabsorb it. So that glucose is going to end up in the urine. And so if there's any excess. So they call that the glucose urea, just the presence of glucose in the urine. And again, that's abnormal because if our kidneys are working properly and we don't have any other health problems, we should have plenty of opportunity to reabsorb all the glucose in the proximal convoluted tubules, and none of it should be in the urine. So with diabetes, that's a condition where you're going to have too much sugar or glucose in your blood, and they call that hyperglycemia. So if you have too much sugar or glucose in your blood, you're going to filter it. So remember, during filtration, glomerular filtration, all of the small molecules and substances in the plasma get filtered and get into the filtrate. So that includes glucose. So all the glucose in the blood is going to enter the filtrate. So if you have hyperglycemia and you have a large amount of glucose, when it enters the filtrate and then moves along to the tubular fluid, if you have too much or you have more than the transport maximum, then you're going to have some glucose that stays in that tubular fluid and makes its way into the urine. The thing with the so that kind of explains why people have glucose in their urine, is because they have overwhelmed their transport proteins. The transport proteins are working as fast as they can and binding all the glucose they can, but there's just too much glucose that outnumbers the transport proteins. So the glucose stays in the tubular fluid and then ends up in the urine. So that is a sign you can see as glucose in the urine. And we in lab, I know we didn't get to do it in lab. I think there might have been a video on it though where you can test the presence of glucose in urine. So you can do that with a kind of a simple little dipstick method, and you can see if there's glucose. So normally never should be glucose in your urine, but if there is glucose, there's a good possibility it could be related to that condition called diabetes mellitus. So these people also have frequent urination and thirst, and so that deals with what glucose is doing with water that is following it, and so that's what results in the frequent urination and thirst. So back to our picture. If we have glucose that stays in our tubular fluid, that makes the solute concentration more high in the tubular fluid. We have glucose in there that's not supposed to be in there as it's moving along down the loop of Henle and distal convoluted tubules. And what it does is it draws in more water, because again, water wants to move by osmosis from an area where there's a low solute concentration to an area where there's a high solute concentration. So as glucose is running through the tubular fluid, water is being by osmosis drawn into that tubular fluid with it. So a lot of people with diabetes mellitus will frequently urinate and then urinate larger amounts because they have more like water in their urine. And then of course they're losing more water that way, so that kind of explains why they want to drink a lot more. So they'll have, be more thirsty and drink a lot more water. Their body's telling them that they need to drink more water to maintain their hydration.